Hey there, and welcome to The Explainer. Today, we are diving deep into one of the most incredible processes in all of life, how our cells multiply. It's this tiny, microscopic ballet that's happening inside you at this very moment, and honestly, it's the reason we're all here. So, let's get right into it.
I mean, really think about it for a second. You get a little paper cut, and a few days later, poof, it's just gone. And you definitely weren't always this size, right? How in the world does that happen? Well, it's not magic, it's biology, and it all boils down to this amazing process of making perfect copies.
You see, the master plan for all of this growth, all of this repair, is packed super tightly inside almost every single one of your cells. And the secret, the absolute heart of this story, is found in your chromosomes. Think of them as the ultimate instruction manual for building, well, you.
So what are these instruction manuals actually made of? To figure that out, we've got to zoom in. No, I mean way in, past the cell itself and into its command center, the nucleus.
Now, inside that nucleus, most of the time, your DNA isn't all neat and tidy. Nope. It's hanging out as something called chromatin, which is basically just this long, tangled-up network of fibers. The best way to picture it, imagine a giant, super messy ball of yarn. That's pretty much your generic code in its everyday, relaxed state.
But here's a twist. Chromatin isn't just DNA. As you can see, it's actually about 40% DNA and a whopping 60% a special kind of protein called histones. And what do? Well, you can think of these histone proteins as tiny little spools. They're absolutely essential for organizing that incredibly long thread of DNA, which is the key to what happens next.
So, how do we get from that messy ball of yarn to a perfectly organized chromosome? Okay, check this out. First, that long DNA strand wraps itself around those little histone spools. Then, this whole string of DNA-wrapped proteins starts to coil up. And then it supercoils on itself again and again, packing an unbelievable amount of information into this tiny, classic X-shaped package we call a chromosome.
And here's what that final structure looks like. Just before a cell gets ready to divide, it makes a perfect copy of all its DNA. This gives us a chromosome made of two identical halves, which we call sister chromatids. And you can see they're pinched together right in the middle at a point called the centromere. They are perfect identical twins.
All right, so the cell has packed all its DNA into these nice, neat chromosomes. It's ready to divide. But hold on, why does it even need to divide in the first place? And what happens before the main event? That all goes down in the prep phase.
The reasons are literally happening inside you right now. First, for growth. I mean, you start as one single cell, and now you have trillions. Second is for replacement, to switch out old, worn-out cells with fresh new ones. And third, for repair, healing those cuts and injuries we talked about.
Now, before any cell can split, it has to get ready. This whole preparation stage is called interphase. You might hear it called the resting stage, but let me tell you, that's totally misleading. This cell is anything but resting. It's super busy growing, making proteins, and this is the most important part, it's duplicating its entire set of DNA to make sure each new cell gets a complete copy of the instruction manual.
Okay, once interphase is done and the DNA is copied, it's time for the main event. This is mitosis, the process where one cell nucleus splits into two perfectly identical nuclei. You can think of it like a beautiful, carefully choreographed four-step dance.
And hey, for anyone studying this for a test, here's a little lifesaver you're gonna want to remember. The 4 stages are prophase, metaphase, anaphase, and telophase. And to remember the order, just think of the magic word, PMAT, P-M-A-T. Let's break down what each letter stands for.
First up, P is for prophase. This is where that loose chromatin we talked about starts to condense and coil up really tightly until the chromosomes become short, thick, and you can actually see them with a microscope. At the same time, the membrane that surrounds the nucleus dissolves, which basically sets the chromosomes free inside the cell. Next up, M is for metaphase. This one's super easy to remember. M is for middle. All the chromosomes, each with their two sister chromatids, line up right in the middle of the cell, along something called the equatorial plane. They're all perfectly lined up like soldiers on parade, ready for the next command.
Then we have A for anaphase. And you can remember this one because the sister chromatids are pulled apart. The centromeres that were holding them together split, and then these special fibers start pulling the chromatids to opposite ends or poles of the cell. It's like a tiny tug-of-war, and as soon as they separate, each chromatid is considered its own full chromosome.
And finally, T is for telophase. The chromosomes arrive at opposite ends of the cell, and they start to uncoil and go back to being chromatin. Then, a brand new nuclear membrane forms around each of the two sets of chromosomes. This finishes the division of the nucleus. And right after this, the rest of the cell, the cytoplasm, splits in two, a process called cytokinesis. And bam, you have two new cells.
So we've just walked through the elegant steps of PMAT. But let's zoom back out for a second. Why is this microscopic dance so incredibly important? Well, the answer is, it's happening everywhere, all the time.
Get ready for this number. Your body, through mitosis, creates about 20 million new red blood cells every single minute, just to replace the old ones that have worn out. That is the incredible power of mitosis in action, constantly working to keep you going.
Okay, so we know mitosis is for growth and repair, and it makes identical cells. But you might have heard of another type of cell division called meiosis, which is all about reproduction. This is a super common point of confusion, so let's just do a quick side-by-side comparison to clear things up.
Let's break this down. Mitosis, that happens in all your regular body cells, your skin, your muscles, everything. Its purpose is growth and repair, and it ends with two identical cells. Meiosis, on the other hand, is special. It only happens in reproductive organs to make gametes, sperm and eggs, and its result is totally different. You get 4 genetically unique cells, each with only half the normal amount of DNA.
All right, pop quiz time. Let's see if you were paying attention. In which phase of mitosis do all the chromosomes line up right in the middle of the cell? Give you a hint. M is for... If you said metaphase, you got it. Awesome.
Okay, question number 2. Let's rewind back to the beginning. What are the two main things that make up chromatin? Remember that chart with the 60-40 split? That's right, it's DNA and those little spools, the histone proteins. Great job.
And now for one final thought to leave you with. We know mitosis makes identical cells. But why is being identical so critical when it comes to, say, healing a cut? Well, think about it. If the new skin cells growing over that wound weren't perfect copies, they wouldn't be able to do their job as skin cells. You need exact replacements to keep your tissues strong and functioning properly. Anything else could lead to problems.
And that is our deep dive into the amazing world of cell division. If you found this explainer helpful and learned something new, please do us a favor and like, share, and subscribe. And definitely don't forget to hit that bell icon so you never miss our next explainer. Thanks so much for watching.